Have you ever wondered how scientists isolate proteins from cells to study diseases, develop medicines, or create diagnostic tests? In today's video, I'll explain what protein extraction is, what protein precipitation means, why they're important, and where they're used in research and industry. Let's get started. Hello everyone, this video is about how to extract protein from cells and how to precipitate it. I already collected cells in 15 milliliters tube by scraping. I centrifuged it to get cell pellets. Put the tubes on ice and add 500 microliters to 100 microliters of lysis buffer. I'm using here RIPA lysis buffer. It will break down cell membrane and cell help to separate proteins from cells. You can also add protease and phosphatase K if you are using protein for immunoprecipitation, but I'm not adding because I will use this protein for simple Western blotting. I added 500 microliters of lysis buffer in my control and desired protein extraction cells tube and collect in Epindrop tubes. Label tubes and put it on ice for at least 30 minutes for complete lysis. Turn on 13,200 revolutions per minute centrifuge machine and set temperature at 4°C and run temperature. After 30 minutes, put Epindrop tubes in centrifuge machine and run it for 20 minutes at 4°C with 132,000 revolutions per minute. After centrifugation, you will see white pellet of cell debris and the upper transparent layer of protein. Transfer protein carefully into new Epindrop tubes and label it and discard pellet. Now time to do protein quantification, because in Western blotting we need to load equal amounts of protein per lane. So for that, we need to know that how much total protein we have in each sample. For protein quantification, I'm using BCA Assay kit. First, I transfer 2 microliters of my protein from each sample to new Epindrop in a 100 microliters of lysis buffer. So it is like 50x dilution of my protein sample. You must have to dilute sample to make it equal to BSA standard, which is important for quantification graph. This is BCA kit in which we normally use reagent A, 1 milliliter, and reagent B, 20 microliters. So in making 2 milliliters solution, so I need to add 40 microliters of reagent B. Mix it properly by vortexing. Now, these are BSA standard, that is starting from 0 microgram per milliliter, 10 microgram per milliliter, 20 microgram per milliliter, 50 microgram per ml. 100 microgram per ml, and 150 microgram per ml. Take 96 well plate and add 25 microliters of BSA standards into each well and then add your sample protein, 25 microliters, and after that add 200 microliters of BCA reagent mixture into each well. After that, incubate in 37°C incubator for 30 minutes. After incubation, read the plated in microplate reader at 562 nanometers absorbance and make the quantification graph. After that, transfer maximum amount of equal protein from each sample into new tube and do protein precipitation step. For protein precipitation, we need ice-cold acetone 4x more than our protein sample and incubate at -20° temperature for at least 2 hours. Or for better precipitation, do incubation for overnight. Next day, take out your samples and do centrifugation for 20 minutes at 4°C temperature with 13,200 revolutions per minute. After centrifugation, remove the upper layer acetone and get precipitated pellet of your proteins. Now add lysis buffer and 5x sample buffer into your sample, and it's ready for running on gel for using as Western blotting experiment. Always keep in mind that after removing acetone, do some air dry for just 30 minutes to remove residual acetone, and make sure to first add lysis buffer, break the pellet, and then 5x sample buffer and put it in heat block for 5 minutes at 95 degrees. After heating or denaturing protein with 5x SDS sample buffer, put sample on ice for 5 minutes directly. Sample are ready. I hope this video will help you in protein extraction and precipitation. Like my video and follow for more.